All right, we're going to go ahead and get started for today. It is my greatest pleasure to introduce Jason Yang. He is a PhD candidate at CalTech currently, and he is co-advised by Yi-Sung Yu as well as Francis Arnold. And today he will be talking about mapping the universe of enzymatic function. Please don't forget to drop your questions throughout the talk in our chat, and Xianyu will help answer those with Jason at the end of the talk today, and you'll also be able to unmute and ask Jason questions. So Jason, we're super excited to have you, and please take it away.
Yeah, thanks, Meg, for the introduction. As Meg said, today I will be presenting my work on the CARE benchmark suite that we recently published on arXiv. But I kind of want to tell this more as a story of how it's really a journey to map the universe of enzymatic function and really understand the world of enzymes and what they do. So to begin, I'll just talk about how most of you know this, but nature has explored a network of evolutionarily connected enzymes. And so these enzymes power everything from essential metabolic pathways that's essential for life to synthesizing other compounds and other natural products. And nature has explored this through natural evolution. So these are all kind of connected on this concept of a phylogenetic tree. But there's really so much function out there that is yet to be discovered. Our picture of enzymatic function looks kind of, if we were to say that this is the universe of possible enzymatic function, looks like this because we have ability to sequence a lot of genes and to determine those that are enzymes. And so we know that over time, UniRef and UniProt specifically have grown significantly. So now there are, for example, over 50 million clusters at 50% identity of sequences. But since we've only been able to sequence so many of the real enzymes that are out there in nature, our picture of the enzymatic universe looks maybe more like this, where a lot of the branches are covered up. And to make matters even more complicated, even though we have all of these genes and sequences, most of them are actually not really annotated for function. So for example, the SwissProt database, which is the expertly annotated and human-reviewed portion of UniProt, is basically less than 1% of all protein sequences that we've been able to actually get the sequence of. So in reality, our picture of the universe of enzymatic function looks more like this, and probably even less so, where there's only a few branches where we understand and we have annotated function. But knowing the function associated with an enzyme sequence is really important for a lot of applications. So just to list a few, one could be metagenomic analysis. So this is where you get metagenomes from bacterial communities or other types of organisms, and then you try to understand the genes in them to understand their specializations within communities. For example, certain microorganisms use nitrogen or fix nitrogen, or other microbes will use alkane as an energy source. Another thing that's important to biologists is finding orphan enzymes, or the concept of knowing the function that's present in nature, but not knowing the specific sequence or enzyme that can do that function. And in broadly, this is kind of categorized under the umbrella term of annotating the functional dark matter. But I think something that's more interesting and maybe directly applicable to engineers is retrobiosynthesis, which is the process of doing chemical synthesis with biocatalysts. So most chemical synthesis is done using precious metal catalysts right now, but there's a lot of interest in replacing these synthesis workflows with enzymes or biocatalysts. So knowing the function of sequences is important for that. And one particular application that's, I think, particularly exciting to me and to my lab is using knowing the function of enzyme sequences for enzyme engineering. And specifically, we do a lot of directed evolution to optimize enzymes for various properties, ranging from thermostability to new to nature activity or other desired objectives. And a lot of my research interests also fall under this kind of optimization process. And we shameless plug, but we have some new work on active learning that just came out. So please check that out if you're interested more in that area. But today I want to talk more about how we actually predict the function of enzymes in a more qualitative way. So before I come back to enzyme engineering, I'll talk a little bit about the tools that have emerged, the computational tools that have emerged to help with these three applications. So for annotated enzymes, it's pretty easy to work with them because a lot of databases have emerged to make it really easy to retrieve these sequences. So for example, one of the most useful ones for retrobiosynthesis is called RetroBioCat. And so in this case, you can search by a target molecule that you want to synthesize using biocatalysts, or you can even search by the enzyme name or the function that you're looking for. And then you can find the sequences that are annotated to do the reactions that you're interested in. And then there's also many protein sequence data sets, such as Brenda and RIA, which are typically connected to enzyme function by an EC number, which I'll explain in a few slides. But generally, this is a way to also search for your function, and then you can link to UniProt, which has sequences. So that's typically the workflow for how you would find an enzyme you're interested in to have a desired function that's annotated. But when for the unannotated enzymes, those ones where maybe you have the sequence, but you don't have the function associated with that sequence, there are also other computational and machine learning models that have emerged to classify enzymes by their function. So the most common one and the one that's still very much the workhorse, I would say, is in bioinformatics, is using sequence similarity. So this would be things like BLAST or hidden Markov models to make alignments and then find the most similar sequence and assign function in that way. And that tends to work pretty well most of the time. But recently, there have been several approaches using deep learning and other machine learning approaches that build off of BLAST or outperform BLAST. And I think one of the strategies that has demonstrated a lot of success in this area is supervised contrastive learning. And so there's been a lot of work that's emerged in that in the past year or two, specifically things like Clean, Hi-Fi Neural Network, and ENS higher, which all kind of use the principle of learning representations where you align things that have the same function and you try to contrast things that have different functions. And that has seemed to work really well. And then there's also been other approaches that have come out. ProtX is very recent, and it kind of combines this sequence similarity-based retrieval with machine learning. And there are many more from the past as well, but I'm just mentioning some of the state-of-the-art approaches here. In general, these models classify enzyme function by their EC number. And so I'll just describe this a little bit in depth because it's important for the care benchmarks in general. But the enzyme commission or EC number is this hierarchical classification scheme for a specific enzyme function that has 4 levels of increasing specificity. And so at the first level, the broadest category, I've listed some of the broad categories of enzyme function here. There's 7 categories. So for example, category 1 refers to enzymes that perform oxidations or reductions. And category 2 refers to enzymes that transfer functional groups and so on. And in this particular example, this is one particular EC class, 1.1.1.1, which is for alcohol dehydrogenase. The 1 here stands for oxidoreductase, so it does oxidation or reduction. The second one here, the more detailed level of the hierarchy, refers to the fact that this enzyme operates on the CHOH group of donors. And then more specifically, it uses NADP as an acceptor. And then specifically, it operates on alcohols, so it's an alcohol dehydrogenase. And so you can see that the EC number, you can kind of think of the EC number as being associated with the main reaction that is performed by that enzyme family or that enzyme function. So in this case, it's oxidizing alcohols to aldehydes with NADPH. And while there are all of these methods like Clean and others that can classify enzymes by their EC number, really there are, I would say, a lack of standardized benchmarks for really evaluating the ability of these models to generalize. And so the way that people have been doing it in most papers that I've seen is, and for this I think was started with the clean paper, is using two data sets. The first one is called Price 149, which is a collection of enzymes that were basically they went back to bacterial genomes and restudied some of the genes and found that some of them were misannotated in this paper. And so this is a collection of 149 enzymes with EC numbers that were previously misannotated and then corrected in this paper from 2018. And the second data set that they use to test the generalizability or evaluate models is called new 392. And this is simply a collection of 392 enzyme sequences that were added to SwissProt, which is the expert validated part of UniProt, after the model was trained. So I think maybe one of the shortcomings of this test set is that it's not really engineered in a way to have to evaluate real world, to basically reflect what kinds of generalization we want in the real world in terms of having maybe different types of, it's not considered like the different types of functions that are available in this new data set, and also the distribution of how similar the sequences are to the training set and so on. So I think this was one of the motivations for why we realized that there's a need for maybe more standardized benchmarks in this area. And then I also want to motivate another need that we found, and this might take a little bit more explanation, but it's one that's very close to my interests in enzyme engineering. But there's also a need basically to find for models, machine learning models that can generalize to reactions beyond those that are categorized under the EC numbers. And our lab is particularly interested in that, which is this concept of evolving new to nature activity. And I think there's a lot of applications for that. I'll list a few here. You can think of CRISPR-Cas or gene editing enzymes as very useful enzymes that we might want to engineer. But in particular, I think for new to nature activity, there are two really big use cases that we are interested in, and I think a lot of people are interested in. One being environmental remediation. So that while there are natural enzymes that can degrade plastics, we're interested in enzymes that can degrade more complicated or difficult pollutants, and that means finding enzymes that can do things beyond what they do in nature. And recently, we were able to find an enzyme and engineer it to degrade silicon carbon bonds. And then thirdly, I think for chemical synthesis, it's really interesting to be able to engineer enzymes to do new to nature reactions. For example, we've been able to engineer heme enzymes to do many carbene and nitrene transfers, and many other enzymes have the potential to synthesize molecules that they don't usually synthesize, such as non-canonical amino acids and so on. So those are all really interesting applications, but it's a really tough process. And one of the things that we've encountered in our lab, one of the bottlenecks for this process is discovering enzymes with new to nature activity, or these starting points for directed evolution and engineering, is a pretty laborious, inefficient, and often ineffective process. So I'll just give you some context on why that is here. So for example, let's say you want to engineer an enzyme to do a new to nature reaction, such as the formation of tyrosine from phenol and serine. So no natural enzyme does this. And this was a project that was pursued in our lab and published this year. But basically, when the lead author on this project wanted to engineer this activity, they couldn't just go to existing databases and you can't just go to like RetroBioCat and find the enzyme that can do this because it's not annotated, it doesn't exist. So what our approach typically is to do this type of new to nature enzyme engineering is to take advantage of enzyme promiscuity, which is the ability of most enzymes to catalyze multiple reactions on different substrates. So for example, you might have an enzyme that's really good at its native activity, but it has very low or initial promiscuous activity for the new to nature reaction that you're interested in. So you can start from there and then further engineer or evolve it towards the new to nature activity. But if you don't know where to start, the kind of the limitations you're facing are that each enzyme you test in the wet lab will take days and also cost hundreds to thousands of dollars. And so you really need to be able to manually filter the database to find the most promising candidates for you to even find your initial activity on. And that often relies on chemical intuition, and it's not always successful. Luckily, in this project, um, the authors had a lot of chemical intuition on how to start this. And so they began with tryptophan synthase, which normally catalyzes the reaction of serine and indole to form tryptophan. And with this, they were able to slowly find initial activity for the desired reaction to form tyrosine. And over several rounds and a lot of time, it was engineered to be able to, using directed evolution, to be able to perform this particular reaction. But just to put this in context, there are many reactions that we would like to catalyze with enzymes for which we have not found initial activity. And so this is a very big challenge in the space. So to summarize, we kind of realized over time from the existing models out there, and also the needs for enzyme engineering, we realized that we need benchmarks to evaluate enzyme function prediction models that are standardized and that apply to real world and interesting generalization problems. And two, we also need a better automated way to retrieve desired enzymes, specifically ones that can generalize to unannotated reactions, which is not something that people have explored much. And so to address one, we developed CARE, which is a data sets and benchmark suite for the classification and retrieval of enzymes. And so this really the goal here was to standardize the evaluation of models that predict enzyme function so that they directly apply to generalization that's interesting in real world applications, such as the ones I described before. And then secondly, we developed a model which uses multimodal contrastive learning called CREEP. And this model is able to perform automated retrieval, and it can generalize outside of annotated reactions. Cool. So I'm going to continue in the rest of the talk by describing CARE and CREEP in more detail. So overall, CARE, you can think of CARE as a framework for evaluating the classification and retrieval of enzymes. So the general process for using CARE starts with choosing the task that you're interested in performing. So we offer two tasks, classification, task 1, and task 2, retrieval. I'll describe that in detail below. But both of these tasks apply to real-world use cases. And then for them, we developed splits for the tasks that directly apply to the real-world generalizations that we're interested in evaluating. And once you've chosen your task and your split, that you can then train your model on the training split and then evaluate on the test split. Task 1 is the one that a lot of existing models have explored before, which is protein to reaction classification. So the question to be answered here is, what does this unannotated enzyme do? And so in this case, you're generalizing to unseen protein sequences and trying to classify the function. And so the general workflow here is you start with a protein, you pass it through your trained model, it predicts the EC numbers, and the EC numbers are then associated with the likely reactions that the enzyme can perform, which describes the function of the enzyme. So this is going from protein to function, or protein to reaction. Task 2 is kind of the inverse direction, which is reaction to protein retrieval. And in this case, the question you're trying to answer is what enzyme can do this unannotated reaction. And the workflow here is the opposite process, where you start with your query reaction, you pass it through your model, and then you get your EC numbers, and that's associated with the likely proteins that will be able to catalyze that reaction. And so the question here really is, or generally the idea here is you're trying to extrapolate or generalize to unseen reactions. So to facilitate the benchmarking of these two processes, CARE consists of two curated data sets called protein to EC and reaction to EC. So protein to EC is a data set that links protein sequences to EC numbers. And this was basically collected from Swissprot with some filtering applied. But for example, if you have an EC number here, it's associated with a collection of maybe anywhere from 1 to hundreds or sometimes even thousands of sequences. The second data set that we curated is called reaction to EC, and this connects reactions to EC numbers. And this was basically a compilation of enzyme map and EC react, which are two data sets that have this sort of data. And so, for example, this tryptophan synthase EC number can be associated with these particular sequences and these particular reactions. And so while its main reaction is to catalyze the formation of tryptophan from serine and indole, it can also often work on indole analogs, which have other functional groups attached to them. So the reason that we decided to operate with the EC number as the bridge between protein sequences and reactions was kind of a design choice based on our thinking at this kind of coarse-grained level representation. We did it in this way because while the goal really is to go from protein to reaction directly and then reaction to protein and vice versa, the data available to train models on that type of direct linkage is rather limited. Enzyme map has some of those data points, but we believe that it's also missing quite a few examples where sequences can actually perform that reaction. So for example, enzyme map might be annotating this sequence to perform this particular reaction with this indole analog, but it could actually have the potential to do reactions on other indole analogs that it's not annotated for. So we decided to operate at the coarse grain level where we assume that all sequences belonging to this EC number have the potential to perform all of the reactions associated with any sequence in that EC number. But that's something that I think we're definitely considering and still thinking about as we move forward. But in the end, we had over 100,000 protein sequences, about 4,000 EC numbers, and then 60,000 reactions associated with those EC numbers. Using this data set, we first designed the task 1 splits, where the goal is to evaluate generalization to unseen protein sequences. So for these splits, we chose 4 splits that have differing levels of sequence identity to the training set, so different ranges. So less than 30% will be the hardest. And things with greater than 50% sequence identity are generally considered to be homologous or to have the same function because of sequence similarity. Then we also incorporated the price data set, which is the one that was previously misclassified. And also we incorporated a data set of promiscuous enzymes, which are sequences with multiple EC numbers. And we think those might be particularly interesting for enzyme engineers because those are often the starting points for engineering new to nature activity. What we did with these was also ensure that there was a diverse spread in EC numbers or functionality in each held out test set, because we are interested in seeing if our models can do well at predicting different types of function and not being biased towards certain types rather than others. As a visualization for maybe people who are more visual, this is kind of how you can think about each task 1 split in the context of each other. So let's say our training set was on the left here in sequence to enzyme function space. So the peaks describe areas where there's enzyme function, and then the XY input spaces sequences grouped by how similar they are to each other. So let's say your train set is over on the left, and as you move to the right, we are assuming that your lower sequence identity to the train set, then your less than 30% identity split is going to be really far away from your training set. The price or the misclassified test split is not necessarily very far away. It sometimes is, but it's more so difficult to classify, maybe because the peaks of in function are pretty close to other peaks. And then the promiscuous test split might actually be really similar to your training split, but those types of functionalities might lie between peaks. So there's multiple correct answers there. And we had a few other design choices for these splits, but we wanted to make sure that it was distributed evenly across functionality. So the pie chart here shows the distribution of all EC numbers and categorized by basically the first level. So there's 7 categories. And then also this is the distribution of all protein sequences in protein to EC. So as you can tell, there are a lot of EC numbers in category 1, but there's fewer actual protein sequences in category 1. And then we compared that to the 6 test splits that we designed for task 1. And in general, they look to be similarly distributed in the ways that we wanted, where there's a diverse range of functionality across different EC numbers. And we also checked the sequence identity of each test split in comparison to the training set. And what we found is that these ones that were divided on sequence identity fell into the categories correctly. And the price split is kind of distributed in the blue across different sequence identities, and the promiscuous split in general is very similar to the training set in sequence identity. Once we had these splits, we decided to retrain existing models, mainly clean, because some of the more recent state-of-the-art methods are not open source or available as code yet. We also compared against BLAST, which is one of the strongest baselines, and also used ChatGPT, which thankfully we still have our jobs because it's not very good at doing this right now. But we also compared against ProtInfer, which is, we were not able to retrain yet for this study, but it's also one of the best performing methods. And in general, what we found is that clean does really well. So using that supervised contrastive learning approach works really well. And interestingly, BLAST does pretty well compared to clean, although there could be some different ways to train clean as well to boost the performance there. We decided to measure performance in only accuracy so that the the performance is a little bit more interpretable. But I think what we've seen is if you use other metrics like F1 score and recall and precision, that the trends are the same. And then we also showed the accuracy up to level 4 or up to level 3, up to level 2, and so on, where level 1 accuracy is the easiest. And interestingly, there are some differences in the trends here. For example, clean is more consistent across all levels, whereas BLAST is actually potentially better at finding specifically what you're looking for. So I think what mostly we're seeing that there's maybe work to be done in this kind of low identity space, but existing models are pretty good at this, the high identity sequences. Yeah, so that is task 1. Task 2 is the one where we were really like entering a world where not a lot of people have necessarily thought about this problem yet. So we decided to frame task 2 as a way to evaluate generalization to unseen or unannotated reactions. And we had 3 different levels of difficulty in the test splits, ranging from easy, medium to hard. And I think this is easier to show visually. So this is kind of a visualization of how we hold out sequences, or sorry, how we hold out reactions in each specific split here. So for example, in task 2 for the easy split, you basically have your hierarchy of EC numbers. So you start with your broadest category at level 7, and then you get more specific, and then it branches out and so on. So at level 4, you have typically have multiple reactions associated with the EC number. So in the easy holdout set, we only held out one of the reactions out of the group under that EC for a bunch of reactions scattered across all of functional space. And then we asked the model to be able to try to predict the EC given only the other reactions in the training set. For the medium difficulty holdout set, we held out all of the reactions underneath the node of the EC number at level 4, so this blue here. And that makes it more difficult because it can't see any of the reactions under that EC number, and the goal is still to predict that original EC number. And then for the hard holdout set, we held out at level 3 and held out all of the reactions under that node. So then to predict the EC at level 3 or level 4 would be extremely difficult. We also checked to make sure things were balanced here. So here's the distribution of reactions across all of the EC numbers. And it looks generally similar to the easy, medium, and hard held out reaction sets. And we also checked using a reaction fingerprint method to measure the similarity between each of the test sets and the train set. And we did indeed find that the easy set is much more similar to the train set than the medium and the hard set. So this was a task that is specifically useful for a lot of things such as enzyme engineering and finding enzymes as starting points to engineer new to nature activity and so on. But there's really not much out there on how to actually do this computationally. So we proposed CREEP, which is contrastive reaction enzyme pre-training, as one of the first models that can perform task 2. And the general approach, which is inspired by a lot of other work, is to learn a representation that aligns and contrasts different modalities. So for example, in your anchor example, in this, it could be a let's say tryptophan synthase sequence is associated with a specific reaction, and that's also optionally associated with a description, with textual description. And your goal is to align this with your goal is to align the representations between these modalities if they belong to the same category. So this sequence corresponds to this reaction, and contrast it if it does not belong to the same. So for example, this sequence does not perform this reaction here, which is an aminotransferase, and this sequence, which is the aminotransferase, does not perform this reaction. So you'd want to contrast across categories and align in the same category. Optionally, we also included textual description as a way to align 3 modalities, because we think that with the growing power of language models to do this sort of scientific reasoning, we think that there's a lot of opportunity for all of these models and representations to work together. And what we ended up doing was fine-tuning existing language models so we can leverage information from all of the unlabeled sequences in addition to these labeled sequences. So for example, we fine-tuned PROT5 for the protein sequence encoding, we fine-tuned reaction FP to encode the reaction, and we fine-tuned SciBERT, which is a BERT model that's particularly trained on scientific text. And just a little bit more motivation on CREEP. The reason we did it this way is because we were inspired by ideas from CLIP, which is probably a lot of you have used DALL-E or OpenAI's image generator, and it uses this approach of aligning text with image and then learning that representation for future generation. And there's also a lot of work on this in the protein space. So for example, in ClipZyme is probably the most similar work to our approach. They encoded reactions as graphs and also protein structures as graphs, and then performed contrastive learning on those representations, which may have their own advantages and disadvantages. So I think there's a lot of opportunity to think about how to combine these approaches. And also some of other work in this space includes things like protein BT and others, where they aligned textual description with protein sequences. Another thing I'll motivate for the reason why we are doing training in this particular way is that by doing this kind of contrastive learning training, we're able to take advantage of two important properties in nature, which are convergent evolution and promiscuity. So convergent evolution describes when you have one function but multiple forms. So for example, there are multiple evolutionarily unrelated isoforms of carbonic anhydrases, so they all look different and they're all unrelated in sequence homology, but they all perform the interconversion of carbonic acid with carbon dioxide and water. So this has been found in a lot of different organisms. And the opposite effect is promiscuity, where you have one form but multiple functions. So for example, this is an enzyme we work with a lot in the Arnold lab, which is cytochrome P450. It's a heme enzyme, and it performs oxidations typically on fatty acids natively. So it does it pretty unselectively. So it actually adds these hydroxyl groups at usually the tail end of the fatty acid, but very unselectively. And what we found is that these enzymes can actually work on other substrates and selectively incorporate oxidations, and they can even be slightly mutated to do things like cyclopropionation. But a lot of these reactions follow the same type of mechanism, but they can look very different and also be using different substrates. And so why that's really important for our training set is that these provide meaningful learning signals. So for example, if we had protein family A, B, and C, and then we had reactions types A, B, and C, if there was no promiscuity or convergent evolution, our model would simply learn contrastively to basically put these clusters on top of each other and then separate the clusters from each other. But when we have these types of properties like convergent evolution and promiscuity, we end up building connections where the model has to learn overlaps between sequence and reaction space. So that can provide a more meaningful learning signal than if we had just had those separate categories. So once we pre-train CREEP, we then can use it for automated retrieval. And so this is just an overview of how the downstream retrieval works. The most common use case will be reaction to protein retrieval in our minds. So this is where you start with a query reaction from your test set, and you'll pass it through your fine-tuned reaction encoder. So this is fine-tuned by CREEP. Then you're going to pass all of your reference set sequences that are proteins through your protein encoder, and then look for similarity between these encodings. Then you're going to rank all of those protein sequences by the similarity, and then you're going to average them in the cluster of which EC number they are. And that is how you're going to determine your ranking of EC numbers from best to worst. And then you can do retrieval accuracy metrics on that. Hopefully that wasn't too confusing, but I'm happy to talk more about exactly how that retrieval process looks. You can do the same with textual encoding. And then what we wanted to do was also as a baseline to looks only at reaction similarity. So this is where we evaluate if like having this second modality is really useful. And so in this case, your test set reaction will just be compared to your reference set reactions. And then you'll do the same kind of process to rank the ECs. Really, the goal is to get from your query reaction to the most likely EC, and then you'll be able to measure the accuracy. So that's what we did with CREEP. We did it with and without text. So there's going to be an example with only two modalities, and there's going to be another example with three modalities. And then we also compared against existing models like ClipZyme, which we were not able to retrain, and also the similarity baseline, and also ChatGPT. I will say with everything with an asterisk here might have somewhat inflated performance just because ClipZyme, we were not able to actually retrain the model yet, so we didn't hold out specifically the same testing sequences that we did with Creep. And then for the ChatGPT, it kind of has access to all the information about reactions in EC numbers, so it might do actually surprisingly well at this because does not really have that held out information. So it's not really the best way to measure generalization. But generally what we found is that creep works quite well. It works often similarly to the similarity baseline, but where it really stands out is on those difficult test splits because the similarity baseline is going to have 0% accuracy on those because it has no training set sequences to actually find the correct EC number for because those are held out. But for creep, we saw that interestingly, it was actually able to get pretty high accuracy on up to level 3 and level 4, even though some of the EC numbers were fully held out. So we think it's very promising, but there's certainly a lot of work to be done in this area. And so I'll just end by saying that both Care and Creep are publicly available, and this is definitely still a work in progress. So we would really welcome feedback from the community. And yeah, I think hopefully I would encourage everyone to use their basically benchmark their function prediction models using Care and using those uh, train test splits. And I think together, really, it's a story of how we can really, include improving the CREEP model to really uncover the tree of nature and discover really useful enzymes and to be able to use those downstream. So yeah, I'll just end and thank both of my advisors, Francis and Yisong, and also just the wonderful lab environments they are to work in here at CalTech. And it's been really great experience and just all of my collaborators as well on this project, both current lab members and also past lab members. So yeah, it's been really great and I'd welcome any questions.
Great. Awesome. Thank you so much, Jason, for that great talk. Feel free to unmute to ask any questions you might have or leave them in the chat. Okay, we got one question in the chat. In the future, would you like to classify the preference of an enzyme to perform the forward or backward reaction?
Yeah, I think if I'm understanding the question correctly, it's maybe like because a lot of enzymes perform reversible reactions. So I think the question is wondering whether we want to see if it prefers a certain direction or to try to incorporate that information. That's certainly something that we can include. We decided to just include the main forward reaction in our current training set, but I believe the RIA database has forward and backward examples. But I think for the most part, we're interested in the preferred forward direction. Any other questions? If not, you can feel free to message me on Twitter or email me, and I'd be happy to explain more or get your feedback.
Okay, great. That sounds like a good idea. Yeah, and if anyone has any questions, feel free to follow up. And just for housekeeping, for next week, we have flow matching at the same time on Tuesday. So yeah, with that, thank you very much, Jason, for giving this great talk. And thanks everyone for attending.
Yeah, thanks so much.
Oh, Jason, there's
Yeah,
one more question in the
I
chat.
can
If
stay
you,
on. Yeah, which kind of sequence data can we employ for the downstream tasks? The second part is, do you consider the other performance evaluation metrics for the benchmark and comparison? And 3, for the selected tools for comparison, I think your method is better as it is specifically designed for a specific task. I guess for the first question, the sequence data is I'm not sure I understand exactly, but you're welcome to unmute yourself and ask the question. But I think it's specifically that we're evaluating on care is using the sequence data provided in the train and the test split. And then for the second question, I think we did look at things like F1 score and recall and other evaluation metrics, but those were basically telling the same story as the accuracy, which is why we went with the more interpretable one. But let me know if you would like to further elaborate on your question.
okay, great. Thanks so much, Jason. I guess with that, we can end this week's seminar, and we'll see everyone hopefully next week.
Yeah, thank you.
All right, thanks.
Right.